Good afternoon, everyone, and welcome to the Office of the Chief Scientist Science Seminar Series. For those of you who do not know me, my name is Mark Edwards, Senior Science Advisor with the OCS and your host for today. In the spirit of reconciliation, I'm happy to offer the land acknowledgement today. I respectfully acknowledge that our OCS offices are located on the traditional lands and gathering place for Treaty 6 and 7, Regions 3 and 4 of the Métis Nations of Alberta. These lands are the home of diverse Indigenous peoples, including the Cree, Blackfoot, Nakota Sioux, Iroquois, Dene, Ojibwe, Saulteaux, Anishinaabe, Métis, and Inuit peoples, and many others whose histories, languages, and cultures continue to influence our vibrant communities today. In keeping with this tradition of gathering, these science seminars provide an opportunity for us to get together to exchange knowledge on interesting science happening across Alberta in an open learning and sharing environment. The hope is that through our dialogue today, we are all able to improve our awareness and understanding and put this knowledge into practice to support evidence-informed decision-making wherever that occurs. Today, we welcome Dr. Ellen Macdonald. For the past 30 plus years, Ellen's research has focused on the ecology of northern forests, particularly forest regeneration, stand dynamics, understory plant communities, and relationships among these. She has endeavored to learn from forest responses to natural disturbance and apply these to improved management and restoration of forests. As an applied researcher, she greatly values productive partnerships with industry and government, which help to ensure that research results are applied to practice and policy. She has been recognized for research excellence by receiving scientific achievement awards from both the Canadian Institute of Forestry in 2014 and the International Union of Forest Research Organizations in 2019. She was the 2019 recipient of the University Cup, the University of Alberta's highest honor for excellence in research, teaching, and service. Ellen has been a member of the Science Advisory Panel since 2019 and is currently in her second term, serving as panel chair since April 2022. In this role, she provides independent advice to the Chief Scientist and Government Alberta on Alberta's environmental science program. Over to you, Ellen.
Thank you, Mark. Thanks for the invitation to present at this seminar series. I'm really pleased to be here, and I'm amazed to see this number of attendees. It's really exciting to see so many people interested in this topic. So my pleasure to talk with you today. So as Mark said, I'm going to be talking about trying to answer this question, does forest biodiversity recover following harvesting, and with a focus on the boreal forest. And I want to start by acknowledging the others who played a heavy role in this project: Anne McIntosh, an associate professor at the U of A Augustana campus, Selena Schutt, who is a research assistant working with us on this project, and two people that were postdocs, Sung-Yil Lee, who's now assistant professor at Lakehead University in Thunder Bay, and Samuel Bartels, who's now an assistant professor at the University of Northern British Columbia. So this was a huge team effort, as you'll see. So the outline of my talk today, I'm going to provide some background, talk a lot about the research approach, present as concisely as possible the results, and then end with some conclusions. So background, as we know, the boreal forest is dynamic. It's been exposed for millennia to natural disturbances, including wildfire, insect outbreaks, wind, and these disturbances drive the dynamics and the composition and structure of the forest. So after a disturbance, there's the forest regenerates, goes through different successional developmental stages, and this provides variety of forest on the landscape. So as a result of the disturbance and the subsequent successional processes, plus influences of topography, soils, climate, we get a wide variety of forest composition and ages across the landscape. And it's this variety of forests that provide the template for biodiversity and for ecosystem function, and from these flow all the ecological, economic, and societal values that we desire from our forests. We can imagine post-disturbance dynamics looking something like this. I'll just introduce a couple of concepts that are key here. So if we have something across the y-axis here, ecological condition of some type and time, and we have the system circulating around, it's not stable, it's moving around in some ecological condition, it experiences disturbance and it changes. So the downward slope isn't meant to indicate that it's a lesser or a degraded stage, just a different stage. And the ability of the system to maintain its condition in the face of a disturbance is resistance. So how large or small this change is over time would be an indication of resistance, and so a smaller change would be higher resistance. And then the ability to recover to the pre-disturbance condition following a disturbance is termed as resilience, and I'll be using these terms during the talk today. Ecological theory tells us that after a disturbance, a system can move to a different state like that, and it may exhibit resilience and go back to where it was before, or it might possibly change to a different condition and stay there and not show resilience. So this would be an alternative state or alternative stable state. Okay, so that's natural disturbances. Harvesting disturbances, of course, are another type of disturbances prominent on the landscape, and they're different than natural disturbances in many ways. And I won't go into that. I know we can all imagine that harvesting is a very different disturbance than fire or insect outbreaks. But forests do regenerate after harvesting, and they follow their own pattern of post-harvest regeneration and successional dynamics. And so the question that we were asked by the government of Alberta is to pose whether forests actually return to the pre-disturbance condition after harvesting, and if so, when does this happen? And this is really key knowledge for forest management and conservation planning, for understanding how we should count post-harvest forests on a landscape in terms of the values that they bring and the biodiversity that they might harbor. So, for example, in forming the Alberta Biodiversity Management Framework, this kind of information was really important. And this project really started with the government coming to me and asking me to help answer this question. So our objective in this project was to try and quantify the ecological recovery of biodiversity. So that is recovery towards the mature forest condition or the pre-harvest forest condition in boreal forests after forest harvesting. And we did this in different boreal forest types. So the boreal forest, as I said, has a diversity of forest compositions across the landscape. In the areas that are targeted for forest harvesting, there's three main types. There's deciduous dominated forests, so that's broadleaf trees like aspen. There's coniferous dominated forests, which in Canada are dominated by white spruce or black spruce primarily. And then there's mixtures, stands that are a mixture of the two. So we divide it up into those three main forest types that we find in the boreal. Okay, so to answer this question, we undertook some research, and we decided to take the approach of looking at relative recovery of species composition. So the response variable we were looking at was community composition, which is really just says it's a matrix of abundance of individuals by species in different biotic groups. And we chose this because it's a representation of the biotic community in the forest. There's other metrics that are often used to look at response of forests to disturbance and recovery thereafter, metrics like abundance of species, richness, and diversity. But those metrics are problematic in that they could go up or could go down in response to disturbance, and it really doesn't tell you if you're getting to where you want to in terms of having that biotic community that's associated with the mature forest or the pre-disturbance forest condition. So we chose community composition, and to look at this as a response variable, we used similarity index. So community similarity index, the Chow-Chicard index we used, and we wanted to calculate this between harvested forests and unharvested reference plots, so plots that were mature forest or pre-disturbance forest. And then we also wanted to calculate this among reference plots because we know that unharvested forests on the landscape are not all identical to one another. There's a lot of variation there. So we wanted to capture that by looking at similarity between reference plots, looking at the variation around those two, and then seeing how these changed over time after forest harvesting. So this use of community composition or relative recovery of species composition is based on a paper by Rosendahl in Science Advances 2019. Rosendahl and co-authors took a very similar approach and looked at recovery of tree species composition in tropical forests after shifting agriculture. Okay, so really what we did here, as you can imagine, we have a harvested landscape here and did paired comparisons between a harvest block and a paired comparable unharvested or pre-disturbance forest, looked at that at different points in time, which we can see actually these are regenerating cut blocks here, and then compared that to similarity between pairs of unharvested forest. Okay, so in the end, we would get something like this. We'd have the community similarity along the y-axis between the harvested and the reference and how that changes over time since harvest. And then we compare that to the average similarity for a reference versus reference with variation around that. So this is an example curve of what we might get. We don't really know what kind of curve. We might have got something like this. When we started out, we didn't know what this temporal pattern would look like. But we would say define recovery or recovered at the point when the curve of the similarity between harvested and reference overlaps with the variability around the mean reference versus reference similarity. Okay, so to answer this question, we needed data, and we thought it'd be good to have as much data as possible. So we took a systematic review approach. So we did a systematic review of the literature through searching on databases on a string of keywords to find all the possible papers that we could that would help us answer this question and potentially provide data that we could use for this analysis. So we managed this in a software called Cobidence that the University of Alberta Library has that helps you manage papers in a systematic review. So after this review of the literature on our string of keywords, we identified 1,922 potentially relevant articles. So then those articles were screened on title and abstract by two people independently to say whether they should be included or not. When the two people agreed, we always erred on the side of including that. So that was largely done by myself and Samuel Bartels. So after that process of screening, there were 472 studies that we decided to retain that were relevant to our purposes. So we use these inclusion and eligibility criteria in choosing whether papers should be included or not. So first of all, we wanted studies that had field data, so not modeling data or simulated data. They had real data from field studies. If we came upon a review article, that wouldn't have provided original data, but we checked the references to see if there were other papers that might provide data that we had missed in our searches. Okay, needed to be conducted in the boreal forest. So this included boreal nemoral or hemiboreal forest, so just towards the south of the forest. And boreal nemoral is a very common term to describe wet, snowy boreal forests in Scandinavia. I had to have an intervention of forest harvesting. So this needed to be either clear-cut harvesting, or we did include partial harvesting if there was very low levels of retention. So 10% or less retention, we included those. Not studies that were, for example, salvage logging or included other silvicultural interventions like harvesting followed by burning or something like that. So forest harvesting, any other regular silvicultural applications like planting and whatnot, we didn't exclude for that. Many studies didn't say what kind of subsequent silvicultural interventions there would have been. They had to have data for the comparator, so an unharvested stand that we could use as reference. And they had to report an outcome of the effects of harvesting on forest biodiversity, including plants, mammals, birds, or arthropods at the community level. So studies that were just looking at one species or two species at a time were excluded. We needed a group of species that represented the community in a particular biotic group. Okay, so these remaining 472 articles that we had then underwent full text screening, again by two people independently, to see whether they should be included. Again, if there was a disagreement, we always erred on the side of keeping the paper in. And this was screening to see whether they met those inclusion criteria, whether they provided a clear explanation of the experimental design, they counted for potential confounding effects, they had information on years post-harvest for the harvested stands, and whether they had true replication or not. So in this process, we excluded 151 studies, and in keeping with the principles of a systematic review, we recorded the reason for excluding all those papers so that if anyone would question any bias or errors in terms of excluding studies, we would know. So every paper that was excluded, we would have given a reason. So you can see here, this study was excluded because it didn't have an unharvested reference and control, and we can see this one. We probably should have caught this on the title, is the Black Hills, Wyoming. It's not boreal forest. So then we had 321 articles remaining, and we did another full text screening of these before bothering the authors and asking them for the data. So to make sure that we actually wanted data from this study. At this stage, we excluded a few other articles. There were situations where we had picked up in our search a thesis, and then the thesis had subsequently been published. So we would have excluded the thesis and just gone with the data from the published paper. Occasionally, we discovered at this stage that there was a paper that still didn't meet our inclusion criteria. And sometimes there were studies that reported the same data more than once. So you might have had a paper that published, the first 3 years of response, and then subsequently there'd be a paper that published 10 years of response that included up to 3 years. And so we would always defer to the paper that had the more comprehensive data set. At this stage, we also sometimes added some papers. So when we checked the references cited from the reviewer synthesis papers, we sometimes found papers that we had missed, and so we added them back in. And some occasionally authors suggested, oh, you might be interested in this paper. So sometimes authors that we contacted suggested papers that we should be adding back in. So we ended up seeking data for 371 articles, and we attempted to contact the authors through every possible means that we could think of, emails, Google Scholar, LinkedIn, any way we could to contact them and ask for the data. Through discussions with authors that we contacted, occasionally they offered up some unpublished data sets that we could use as well. What we were asking them for was data on species abundances at the level of the true replicate. If they didn't have it at that level, they give it to us at the level of the subsample, and then we could average it to the level of the true replicate for harvested and comparable unharvested reference stands. And we asked for any data on arthropods, birds, small mammals, vascular plants, bryophytes, and lichens. We also wanted information on metadata about the study, so make sure that we had information on the location, forest type, dominant tree species, and years post-harvest. We had originally hoped to include some other biotic groups, but at the end of the day, we decided there was too few studies to really do a meaningful analysis, so we ended up excluding amphibians, fungi, mollusks, reptiles, soil fauna, and soil microbes. In the end, after a lot of effort and emails back and forth and bugging authors and having them say they'd send it next week and then the week after that and the week after that, we eventually got data from 81 different published studies and 2 unpublished studies. And this resulted in 176 data sets. So some articles provided more than one data set. They might have studied more than one forest type, more than one biotic group, more than one harvesting intervention, say a clear cut, and then a low level of retention. So here's how it stacks up in terms of the number of data sets we got. A pretty equal number of data sets for most groups. Arthropods, we got a lot of data, and we ended up dividing those into some subgroups. Beetles as a general category, carabid beetles, saprozoic beetles, and spiders. Small mammals, we got a slightly less number of data sets, but we decided that was enough to analyze. And this shows how many reps, so the replicates within the data set, the total. So in the end, we had 3,113 comparisons between harvested and reference forests. Where were the data from? Here's the geographic distribution. So a very good distribution throughout the Boreal in Canada from pretty much coast to coast, and a really good representation in Europe as well, particular Scandinavian countries, and some data from Russia as well. So in terms of the data sets, we got more from North America than Europe. Beetles and bryophytes, pretty well equal numbers from both locations. For birds, mammals, spiders, and vascular plants, we had more from North America than from Europe. And for lichens, we had more from Europe than from North America. In terms of the years post-harvest, we were really pleased to have a really good spread. So between 1 and 73 years post-harvest for the studies from Europe and 1 and 80 years post-harvest for the studies from North America. And pretty even distribution among the different biotic groups. A couple of groups, spiders and mammals from Europe, pretty low range in terms of years post-harvest. Saprozoic beetles a little bit lower, but otherwise we had pretty good representation of number of years post-harvest for all the different biotic groups that we looked at. So as we had this data, as you can imagine, there was a lot of work going into data formatting, quality assurance, quality control. We gave each data set a unique code. We had to extract the metadata for each data set, location, forest type, years post-harvest, biotic group, etc. Then we calculated the similarity index. So as I said before, we used the Chow-Jacquard index and calculated pairwise similarity between harvested plots or replicates and the unharvested reference in each data set, and then community similarity among the unharvested reference replicates within a data set. So every data set had to have at least two true replicates of the unharvested reference. Then we analyzed these using generalized additive mixed models. So we were modeling community similarity, the harvested versus reference, as a function of years post-harvest. And we used mixed models, so we included data set as a random term. So this was so that data sets that had a larger number of replicates versus a smaller number of replicates wouldn't be more influential in terms of driving the overall curve in the GAM. So we ran these GAMs for each biotic group separately. We did it first for all the forest types combined with an analysis that included forest type as a predictor variable. So we wanted to see whether there were differences in the response among the forest types. And then on the basis of there being good evidence that forest types did differ, we ended up doing separate analyses for each forest type separately. The important thing about GAMs here is that they allow for nonlinear responses. So there's no dictating what the shape of the response curve will be. They're not confined to be a particular curvilinear response or a linear response. As you'll see, there's a wide variety of nonlinear responses that you can model using a GAM. More on the data analysis, we then calculated the weighted mean similarity for unharvested reference versus reference. So this is the paired reference stands. And so we weighted it. So we calculated the mean within each data set and then the mean of those. Again, so that data sets with more replicates wouldn't be more influential in terms of the overall mean similarity between reference stands. Confidence interval around that. Plotted the GAMs with the trend of similarity versus years post-harvest, plotted the mean similarity among unharvested compartments with confidence interval. Then, as I said, we defined recovery as the point in time when the confidence interval of harvested versus reference curve overlapped with the confidence interval of the reference versus reference being used as a comparator. I know that was a lot. You're going to see a graph in a minute that hopefully makes this clearer. Okay, so some results. So this is an example for all arthropods and for all forest types combined, and then for each of the forest types separately, conifer, mixed, and deciduous. All the data points are the individual data points of the similarity between a harvested replicate and the unharvested reference replicate. The curve here is the output from the generalized additive mixed model, so a curvilinear, as you can see, and the gray is the confidence interval around this. This little teeny dot here with error bars, you can see a little bit better for the deciduous forest here, is the mean similarity between reference compartments with the error bars around it. So this is our comparator, and on the y-axis is Chow-Jacquard similarity. So 0 is no species being shared, and 1 would be essentially identical composition between the harvested stand and the reference stand. And as you can see here, by looking at this confidence interval and going across the curve here, we can see the point in time when there's an overlap between this confidence interval in the recovery curve and the confidence interval of the reference stand versus reference stand, and that would be the number of years post-harvest. So we see here it's just a little bit less than 35 years in this case. Okay, so we looked at all the curves that we got and created these 4 generalized recovery curve types that the different biotic groups and forest types can conform to. So similarity here, years post-harvest, and here's our reference on the right-hand side. So this first one, decline in similarity followed by recovery and then a leveling off at a condition that was overlapping with the reference versus reference. So this really is sort of a lagged resistance followed by resilience. So initially it looks like there's resistance, but then over time we see a lack of resistance with a change from the pre-harvest condition and then followed by resilience up to the point where there's recovery. This curve here really shows resistance, so not much difference, not much change after the disturbance between the pre-disturbance or the unharvested condition and the harvested condition. Then I said variable resilience because there was a number of these with very little change over time, and sometimes they got to the point where they overlapped with the confidence interval of the reference versus reference, and sometimes they never did or they barely did. Curve C here, I just call variable resilience. So it's more or less linear. Sometimes there was a little curvilinear bit at the beginning or at the end. Sometimes it often came within the range of the reference versus reference. Sometimes it leveled off, just barely reaching the condition that we'd call recovered. And then the last one here is just a decline, like it's the lagged resistance here, initially looking at like resistance and then declining resistance over time, and no resilience, no evidence of recovery towards the reference condition. Okay, so I'm going to go through these quickly. I'm going to sum them up later on, but where am I time-wise? Okay, this is good. So looking at conifer, mixed and deciduous forest, here I show the type of curve that we saw followed, and here I have years to recovery. So for example, this is all the arthropods combined. The conifer showed this sort of lagged resistance followed by resilience and about 35 years to recovery. Mixed forest showed more or less a linear pattern of resilience with about 20 years to recovery, and deciduous forest again showed this lagged resistance followed by recovery with about 15 years to recovery. Okay, then we broke down the arthropods into some different groups. So all beetles combined, the pattern follows the same as for all arthropods. Similar times to recovery, except for that the mixed, it was really low. So it was like less than 5 years to get into the overlap zone with the reference versus reference for just the beetles. Then specific groups of beetles, so carabid beetles, these are ground beetles. This again follows the same curve as for the all arthropods with 35 years of recovery. Deciduous forest follows the same pattern as all arthropods and beetles with 15 years of recovery. Here, carabid beetles followed the curve that showed a lack of resistance and resilience, and all I could say was more than 15 years to recovery. And we'll see again why that was, is because 15 years was the longest time span for mixed forests with carabid beetle data sets. So that's the maximum times post-harvest that we had data for. Saproxylic beetles. So saproxylic beetles are beetles that are associated with deadwood. We only had data for them from conifer and mixed forests. Again, the conifer forest showed the same pattern, lagged resistance followed by resilience, and I could only just say over 15 years till recovery, and mixed forest had the same, just sometime greater than 25 years. So that's a reflection of the maximum times post-harvest that we had data for. And finally, we have spiders for conifer and mixed forest, a linear response of resilience. For conifer forest, about 25 years post-harvest to recovery. For mixed forest, something less than 5. And for deciduous forest, the lagged resistance followed by resilience with something less than 5 years time to recovery. Okay, moving on from the bugs to the birds. So this is songbird data. Conifer forests again showed this same lagged resistance resilience with about 35 years post-harvest being the time to recovery. Mixed forest and deciduous forest both had the more or less linear response. And for mixed forests, about 35 years post-harvest to recovery, and deciduous 15 years. Small mammals, we mainly had data from conifer forests. They showed resistance, so the bee pattern, so very little change after the disturbance from the unharvested condition, and recovery in about 25 years. We only had three data sets from mixed forest, but I just wanted to show you if we added those data sets in with the conifer and looked at the pattern for all forests, it showed the same pattern. But because those three data sets showed more like the D pattern or the lack of resilience and resistance, the number of years to recovery increased to something greater than 55 years. Vascular plants, my favorite. Same pattern in the conifer forest again, about 30 years post-harvest. Same lagged resistance and resilience in mixed forests, about 15 years post-harvest. And deciduous forests, a more or less linear response with about 10 years post-harvest. Bryophytes, so this is mosses and liverworts. Interestingly, in the conifer forest, it showed more or less a resistance, so very little change from the difference between the harvested and the unharvested, but poor resilience. So it was about 75 years before there was an overlap between the harvest and unharvested reference similarity and the reference unharvested versus unharvested. Same pattern for mixed and deciduous. All of these shows this resistance, but with poorer resilience. For mixed forests, about 25 years to recovery, and same for deciduous forests. Finally, lichens, we only had data from conifer and mixed forests. For the conifer forest, more or less a linear response, although relatively longer time period to recovery of about 50 years. And for the mixed stands, again, this pattern that showed a lack of resistance and resilience, and could only say that it was more than 10 years to recovery. And again, that's because about 10 years post-harvest is the longest time period we had data for lichens in mixed forests. Okay, if we combine these two together again in one analysis, we get the same linear response, but because of the influence of the data from the mixed forest, the time period to recovery goes up to something greater than 70 years for lichens. Okay, so to summarize all that information, the most common pattern we saw was this one. So this sort of resistance lag followed by resilience. So the forest initially didn't change that much. It took a few years before we saw the difference between the harvested and the unharvested after the harvesting had occurred. This wasn't particularly unexpected. This is a reflection of ecological inertia or ecosystem memory, where it takes a while for the biota to respond to the new forest conditions associated with harvesting, and then a curvilinear response leveling off to a recovered condition showing resilience. Next most common pattern was this one, sort of the variable resilience, more or less a linear response, maybe with a little curvilinear at one end or the other, but eventually reaching the recovered condition. And then the two less common patterns were this one, the resistance pattern, so very little change versus the unharvested condition, but not necessarily achieving resilience by the end of the time period. And then finally, this pattern with low resistance and resilience with a decline in resistance, and then no evidence of recovery over time. The important thing to note about this pattern D, we only saw this in two data sets. And those were data sets where we had a very short time period post-harvest. So this may well be, this D pattern may well really just be the start of the A pattern, and we never got to the time period where it turns around and moves towards recovery. We really can't say because we had a short time period post-harvest for the data sets where we saw this pattern. Okay, so to summarize, as you saw, and maybe somewhat surprisingly, many of the biotic groups had recovered within 35 years or less, so had a similarity to the unharvested forest that was within the same range as we see between two comparable unharvested forests. Deciduous and mixed forests tended to recover faster than conifer forests. You may have noticed that as I went through the results. So for conifer, the results were something, the low end was something greater than 15 years and going up to 75 years for recovery post-harvest. And in comparison, for the mixed forest, sometimes it was as little or less than 5 years to recovery, going up to something that was saying something greater than 25 years or actually at 55 years for recovery. And for deciduous forests, again, the low end was something less than 5 years to recovery, going up to 25 years. And so I think that this makes sense knowing what the ecology of these forests is. So a conifer forests tend to take longer to develop. They tend to be a later successional stage in the mixed wood. And you can imagine that because of the heavy cover of conifer trees, that a harvesting condition is changing those conditions much more dramatically than it would in a deciduous forest. A deciduous forest, on the other hand, loses its leaves during part of the year. It's more an early successional kind of forest. The deciduous trees, the dominant deciduous trees, aspen and poplar, both in North America and Europe, recover quite quickly, regenerate quite quickly after disturbance. And the contrast in the ecological conditions, the climate conditions in the forest after harvesting are less for deciduous than they would be for conifer. So this difference makes sense ecologically. Despite the fact that many of these groups recovered within 35 years, there was some notable exceptions. So poorer recovery, for example, for bryophytes being about 75 years in conifer forests, lichens 50 years in conifer forests, or perhaps more than 70 when we look at the results from the all forest analysis. And maybe for small mammals, it was 25 years in conifer, but then when we added in the mixed wood results, it jumped up to something greater than 55 years. So perhaps for small mammals. So those groups, particularly the bryophytes and lichens, again, this makes sense. They're species that are associated, they grow, a large part of the biodiversity is growing on dead wood. They're very dependent on moist microclimates, good cover of the canopy to support them. And so it makes sense, again, that they would have less resilience than other biotic groups. Okay, so the conclusions from this. The conclusion seems to be that the boreal forests show relatively high resistance and or resistance to harvesting disturbance. But there are some important caveats. So conifer forests seem to have lower resilience or resistance than mixed or deciduous forests, and this makes sense for the reasons that I just explained. And species groups that are associated with mature forest conditions or closed canopy conditions showed lower resistance and resilience. And this was really notable for the data from the bryophytes and the lichens. Caveats. Please don't leave this seminar saying boreal forests are completely resilient to harvesting and we don't need to worry about it. Again, this was a community composition, so it's representing the whole suite of species. The Chow-Jacquard index is one that we chose because it's particularly better at dealing with uncommon or rarely observed species. But there still could be uncommon species, and particularly species that are particularly sensitive to harvesting, and we didn't look at the responses of them particularly or specifically. So certainly there are species that will still have negative impacts and long-term negative impacts of harvesting on their abundance, and we didn't look at that specifically. And of course, there's lots of biotic groups that we didn't consider. So we didn't consider large mammals, we didn't consider birds of prey. As I said, we weren't able to consider amphibians or reptiles. So there are a number of biotic groups that we didn't represent here, and particularly those requiring large continuous habitat, animals that move across the landscape at a much larger scale, animals or other biota that are dependent on landscape connectivity. That scale is not what we were looking at, so we can't say how they would respond. The other important thing is the influence of the studies with the longer years post-harvest. So we did have, as I showed, a really nice representation of range of years post-harvest, but not all studies certainly had the longer time periods post-harvest. And so the endpoints of the curves that we showed are going to be influenced by those studies that did provide data for the longer time periods post-harvest. And so we need to think about where those studies are located, what the harvesting practices were. If you're studying a forest that was harvested 50, 60, 70, even up to 80 years ago, certainly there's been changes in harvesting practices over time. They may have been less intensive than they are now. They may have been more intensive than they are now. So we need to think about the influence of those studies that are longer time periods post-harvest, because they're particularly influential in driving the endpoints of those curves, which is going to determine the point at which we say there's recovery of the forest. The other thing that is really important to notice here is, even though we have a nice curve with a nice error bars around it because we had a ton of data to work with, there's huge variation in the post-harvest forest composition. So we see some instances where even shortly after disturbance, the forest is essentially identical to unharvested forest. And at the same time, we see time periods a long time after harvesting where many forest stands still showed substantial differences in comparison to the unharvested forest. So although the curve shows here, there is a large proportion of the data that's falling down here. And so we can't forget those stands, though, that not every stand is going to follow this curve. And so we need to consider the possibility that certain stands still are going to be dramatically different than the unharvested condition many years after harvesting. So where do we go from here? Possibilities for future research. Examine the effects of variable retention levels. We are doing this with data from the EMEN project in Alberta. This example of variable retention harvesting in the bottom here. What I think is really interesting is to explore the factors that are driving this variation in recovery. And I think the recovery of the forest and the way it regenerates and what composition it has is particularly influential, obviously, in driving the biota afterwards, and could provide some important insight into what's driving these different time periods to recovery. And so if we look at the influence of post-harvest regeneration and the composition of the forest afterwards, I think it'll provide some additional insight, in particular into that spread of points that we see for the longer time periods post-harvest. Okay, I want to thank the funders of this research. So we received funding from Alberta Biodiversity Monitoring Institute, matched that with money from the MyTax program, additional funding from Alberta Conservation Association, and some funding from the University of Alberta. I used my research funding that I got as chair of the department to support this project. I want to thank Michelle Rude, who was a research assistant on the project early on. I want to particularly thank all the researchers who shared their data sets with us. We were really gratified with the positive responses that we got, and all calling upon our friends and colleagues around the world to share their data, which is not always easy to do. Dig up old data and try and get it formatted and send it to somebody. So people were really very generous in sharing their data with us. And of course, all those data originally were collected by many field assistants, and we're incredibly grateful to them for collecting the data that we used in this study.
Okay, thanks everyone for joining our science seminar today. Our next science seminar will be on March 14th, and we'll be hosting Dr. David Olefeld from the University of Alberta, who will be presenting on climate change and peatland functions in Alberta. Hope you can join us. As a reminder, if you're not on our mailing list, to receive invites to our science seminars and would like to be added, or you and your team have work that you would like to share at an upcoming science seminar, please contact the Office of the Chief Scientist at aep.ocs.gov.ab.ca. We'll end there for today. Thanks again, Alan, for the great presentation, really interesting. And to everyone who tuned in, we'll see you next time. Thank you so much.
Thank you so much. Bye.
Okay, bye.